The goal of our team is to understand the organization of motor circuits underlying movement and locomotion. We want to establish how each neuron of these networks are dynamically active to enable locomotion. We focus our approach on a small, transparent vertebrate, the zebrafish larva. Most of our experiments are performed on living animals while they are moving. We develop optical techniques to monitor neuronal activity while the animal is in motion. Because the larva is transparent, we can use a technique called optogenetics to target specific neurons and test how they contribute to the generation of behavior. We can also transform the activity of neurons into light in the microscope. We study science because of a sudden moment of emotion. For me, this emotion came when I realized that we could read, without sacrificing at all the animal, the activity of neurons in the brain and in the spinal cord in order to understand the neuronal code via the light emitted by neurons. This, from the very first moment, fascinated me. We want to understand an enigma, namely how sensory information is integrated into movement. To tackle this question, we split the work within the team. Some of us work on sensory information, some of us work on the analysis of movement, while others establish the connectivity pattern of neurons in the motor circuits. We had to combine genetics, behavior, optogenetics to manipulate the elements of the circuits. We became interdependent as a group, and we learn a lot from each other because we look at different elements of the same structure from just different angles. Each of us came with her or his expertise and learned new techniques in order to develop together an interdisciplinary project. Moreover, the projects are so interdependent that one result from one project enlightened the strategy to adapt for the other ones. I love this dynamic approach of science. I think it's essential and precious to form a link between people because we are smarter as a team. Our team research is mainly fundamental at this stage. Our goal is to learn in a simple animal model the principle of organization and function of the spinal cord in order to help us design a strategy to initiate locomotion in humans in the future. Working on a hospital campus enables us to bridge with medical doctors who have different training and knowledge that are very complementary to mine. The interaction happens both ways. One point that we all find important is that the brain triggers a movement, but the real motor lies in the spinal cord itself. And it is there and relatively intact in spinal cord injury patients. And the spinal cord is still there and relatively intact in patients after injury. So it's very promising that evidence from humans and animals indicate that for paraplegia, if we manage to stimulate the spinal cord via sensory pathway, we can achieve postural control and the beginning of voluntary movement. Understanding these points in order to improve the life of people suffering from injury would be astonishing.